Hi, really excited to welcome you all to our Neurodegeneration Early Career Research Meeting 2021. I hope some of you are returning from the one that we did last year. It was a really huge success and I'm so excited to present this to you all today. And so just briefly, the agenda is we'll start with an introduction from myself. So I work for Proteintech, and then also from Sarah, and Sarah will be our chair, and I'll introduce Sarah in a second. So we're going to start off with our keynote speaker, and this is Dr. Shane Liddlow from NYU Langone. All these times on the left are in Central Time US. And then we're going to have 5 of our amazing ECRs. And yeah, please do note the times, and yeah, please stay as much as you can. But this will be recorded because I understand that most of you will probably have other activities to get to, such as working in the lab. But yeah, this will be recorded and will be online later. So I'll definitely be sending this all out to you afterwards. And we also have a Neuro Week going on at the moment. And we have our really popular tote bags and other things and giveaways of free antibody samples. So I'll make sure to let you all know how to access those as well afterwards. So I'm just going to introduce our wonderful chair. So our chair today is Dr. Sarah Ryan. And Sarah Ryan is an Alzheimer's Society Research Fellow, currently working on frontal temporal dementia and motor neuron disease at the University of Manchester. Previously, Sarah completed her PhD and postdoc training at the University of Manchester, and this was after an MSc in Neuroscience at King's College in London and a BSc at the University of Leeds. Her research focuses on the role of microglia and inflammation in disease. And now I'm going to stop sharing my slide and hand over to Sarah.
Thanks, Becca, for the really nice introduction, and thanks for having me today. I came to the event last year and it was excellent, so I'm really excited to be a part of it today. I've read all the abstracts, so I know we've got a fantastic lineup. We're going to have some really good science and hopefully some really interesting chats. So welcome to everybody joining from home. I hope that you're all going to get involved as well. We'd really like this to be as interactive as possible. So please do, as the speakers are talking, if you have questions, type them into the chat box, and I'll read out as many of the questions as we can after the talks and get as many in as we can. Any questions that go unanswered, we'll try and keep the conversation going over email or Twitter afterwards as well. We also have a Twitter hashtag for those of you who like to tweet. So if you can tweet with hashtag PTG Neuro21, you can all join in the conversation and find each other on Twitter. So enough from me. I'm going to get started introducing our first speaker, which is our keynote, Dr. Shane Liddlow from New York University. So Shane did his undergraduate degree and PhD at the University of Melbourne before moving to the US for a postdoc with Professor Ben Barrows at Stanford University. During his postdoc, he became interested in astrocytes and made important discoveries about the links between astrocytes, microglia, and neuronal function. He's previously won a number of prestigious awards, including the NHMRC Australia CJ Martin Training Award and the Glenn Foundation Award for Aging. And he was also named a Stat News Wunderkind in 2017. He's now an associate professor in the Neuroscience Institute at New York University Grossman School of Medicine, where his research continues to focus on astrocyte function in health and disease. But rather than me talking for too long, I'll let him tell you all about that himself. So without further ado, over to you, Dr. Shane Liddlow.
Well, thank you very much for that extremely kind introduction, and hopefully everyone can see my slides and hear my voice. If not, I will continue to make noise into the void, and we'll see how many people angrily tweet at me. You know, I tend to try and say no to a lot of speaking invitations because it takes me away from interacting with those amazing trainees in my lab and doing all the exciting science that's going on. But whenever I'm invited either by trainees or for a conference that is essentially for trainees, I find it really hard to say no. And so today is a perfect example of giving trainees the platform and the voice to talk about your amazing science. But what I want to talk about today is maybe a little bit of a rambling story. As you heard, of course, you know, my research interests are in astrocytes and glia and how they contribute to neurodegenerative diseases. But also I'm a pretty staunch advocate of making sure that trainees have the right support that they need from a scientific sense and and from an administrative sense to make sure that they can be the best people that they can be. And so I thought it would be fun to sort of weave together the support in both the science side of things that we do looking at Glia and also the side of things that you need academically, which is mentors. So, you know, I know that this is a very much a simplification of the way the world works. It is not all black and white. There's obviously a lot of gray in between. But on a broad sense, we like to think of life and science falling into two categories. Either there are cells or people around who are helpful, or there are cells and people who are around who are harmful. And, you know, from a research side of things, we like to think of glia being sort of these support cells in the central nervous system. They essentially, they're not electrical output cells, but they make sure that neurons can do their job so you can live and laugh and love and feel and all those sorts of things. And without the glial support, particularly what my lab is interested in, the astrocytic support, your brain doesn't work properly. And so on the helpful sense, they're very important to have. But of course, on the harmful side of things, if anyone's familiar with the work that's coming out of my lab in the last few years, we're really interested in neurotoxic reactive astrocytes, or astrocytes that, even if they're not actively killing neurons, may passively just simply give up on their tasks at hand and leave those neurons to die. But of course, this dichotomy of helpful and harmful can also be applied to mentors. You know, a very helpful mentor will support their trainees and make sure that they can be the best version of themselves. And even if you aren't aware of what they're doing, they're in the background putting things in place to make sure that you are put your name up for awards, make sure that you're getting exceptionally strong recommendation letters. And of course, although this is the vast majority of mentors who are there for the betterment of their trainees, there are, of course, some harmful activities that can happen where mentors may actively give you no support or be extremely aggressive and mistreat their trainees. So I sort of want to keep this dichotomy up throughout the remainder of the talk. And because I'm a child of the 80s and I really love B-grade horror movies, I thought that a nice way to sort of visualize this would be using the Mogwai and the Gremlins from the late 80s movie Gremlins. And so what we want to do is essentially have these extremely helpful glia and mentors in your life and not have this parochial, hierarchical, cigar-smoking monsters around in our lives. So what I thought we would do is continue this sort of dichotomy while we talk a little bit about science and some of the exciting work that we're doing that may be of interest to some of you. And I'm happy to talk about that offline if anyone has any deeper questions. But I really wanted to spend a considerable amount of time just talking about life and my trajectory so far and how mentors have been an important part of that. And so to that end, I really wanted to start, and this is a rule that we have in my lab, that we always start by acknowledging all of the people that have actually done the work that I'm going to present. One of my biggest pet peeves and hates that I have is when mentors or people, PIs, present the work and then they rush through their acknowledgements at the end of the slide. So everyone who's highlighted here in yellow has actively contributed to the work that I'm going to tell you about today, even if it was only in a small way. The main stories that I'm going to talk about, though, were spearheaded by Kevin Gutenplan, who was a graduate student who I was very fortunate enough to work with while I was in Ben Barris's lab at Stanford. And after Ben's passing in 2017, Kevin became sort of an adjunct member of my lab here at NYU. He's now a postdoc in Mark Freeman's lab at the Bollum Institute. And I'll also spend some time talking about some work from Philip Hazel, who's a postdoc in my lab, who's been spearheading some really exciting work into the heterogeneity of astrocytes in the context of disease. And he's been ably helped by Michael O'Day, another graduate student in the lab. So for those of you who don't know, I'm going to give you Astrocytes 101, a very, very short overview of what we think they do. And the reason that this is really important is that at heart, my lab is a cell biological lab. We look at how cells interact with other cells, and we ask the question, is it the same as what happens developmentally or physiologically, or is it different in the context of pathology? And if we understand how these functions change, we envisage that we can understand how there may be some perturbations or some contributions to the degeneration of neurons in chronic neurodegenerative disease. So we know, like good supporting mentors, that astrocytes keep neurons alive either by providing them with nutrients or taking away metabolic rubbish molecules. We also know they're important for the maturation and formation of synapses during development, and they have really important roles that involve interactions with immune cells, either centrally, that is microglia, or peripherally if they're diapedesing across the blood-brain barrier. And one can imagine that a change in any one of these functions could actually be manifested in neurodegenerative diseases. For instance, we know in Alzheimer's disease, very early and in the prodromal stages, there is a loss of synapse density. And this was shown really nicely by Su-Nara Hong when she was in Beth Stevens' lab at MGH. But of course, there's the opportunity in disease or following insult that astrocytes can become what we would call reactive. That is, they are reacting to an external stimuli. And so maybe they become less efficient at the task they were doing before. So maybe it's not that they don't provide any metabolic support, but maybe they just don't provide enough metabolic support. And so neurons begin to starve and die. Or maybe they don't properly recycle neurotransmitters, and then neurons can become excitotoxic and die. There's also some subtypes or subtypes of reactive astrocytes that can re-enter the cell cycle, become extremely proliferative, and actually form borders or scars around any invading pathogen, or in the instance of AD, they could form this around an amyloid plaque. And what we are particularly interested in is the fact that these astrocytes may gain functions that they didn't have beforehand, and they could actively start secreting molecules that could contribute to the death of neurons in very specific circumstances. So this, of course, is not to say this is all of the functions that astrocytes have, but this is the number that we have really good in vitro and in vivo assays for, and we turn back to time and time again. So how do we like to study this? Well, we're really interested in the heterogeneous profile, functionally, transcriptomically, proteomically that these cells have. But of course, the way that we do this is, of course, we have to start with transcriptomic data. And the reason we say we have to is because by and far it is the most cost-effective and readily available way for us to understand this. It used to be that we'd do this by sorting astrocytes and doing bulk RNA sequencing. Now we do this with single-cell RNA sequencing and spatial transcriptomics. And with that, we identify transcriptomically unique subpopulations of these astrocytes, and then we work out ways of recapitulating those subtypes in a cell culture model so we can actually study their very specific interactions between other central nervous system cells, be they neurons, microglia, endothelial cells, etc. And the reason we do this in addition is twofold. One, we have extreme control. It's very hard to do a lot of these assays in the first instance with any degree of high throughput approaches in vivo, although they do get validated in vivo. But ultimately, we're interested in how we may actually target these subtypes for novel therapeutics. And so we need to have a cell-based assay so that we can do high throughput screening. And then, of course, we like to overlay onto that all the complexities that come with life that are apparent when you have chronic neurodegenerative diseases in humans that last for several decades. And so we can look at whether or not there's any differences across the age of the individual, the brain region those cells are from, sex, or even any underlying disease etiology, such as mutations, or in the context of astrocytes, maybe different isotopes of activity. So just sort of a very quick overview. A few years ago, when I was a postdoc at Stanford, I was able to discover that there was a subtype of reactive astrocytes, this neurotoxic subtype of active astrocytes, that transformed from a physiologically normal and very helpful cell to a neurotoxic phenotype, and was able to determine exactly what cytokines were required to induce this. And this was really nice because we knew what we could actually recapitulate this. In addition, we could use some markers such as the complement component C3 to localize those neurotoxic reactive astrocytes in regions of the human postmortem brain that had undergone degeneration. But ultimately, what it enabled us to do was do this functional testing. Is there something the astrocyte did before that they no longer do? And is there something they do now that they didn't do before that can help us understand what's happening in disease? And so if we go back to our wheel of functions, some examples of what these astrocytes do in this substate is that they fail to promote the formation of structural and functional synapses. This could be really detrimental, like we mentioned in the prodromal stages of AD, but also they almost completely shut down their phagocytic machinery, which could be really important in removing cellular debris, amyloid plaques, etc. And as I sort of alluded to, the one function we found that these astrocytes do have that they didn't have before was that they secrete a neurotoxin that is toxic to mature neurons and mature oligodendrocytes, but not to any other cells in the central nervous system. And I won't talk any more about this toxin, but in some follow-up work that we did with Kevin Gutenplan, he was actually able to discover that it's a fully saturated long-chain free fatty acid or a fully saturated lipid that is secreted by these astrocytes that is potently able to cause the death of these neurons. And this was published a few weeks ago in Nature. Now, one of the sort of caveats or the niggling doubts that we always had in our mind with this work was that we did all of this using rodent cells, and we did all this in vivo in rats and mice. And so while we were able to at least correlatively show that these cells were present in regions of neurodegeneration in human brains, we never knew if the functional changes were the same in human astrocytes. And so when I moved my group to NYU, we partnered with a very good friend and collaborator, Valentina Fossati at the New York Stem Cell Foundation, and two amazing researchers spearheaded this project. Lily Babar, who was a research associate in Valentina's lab. She's now an MD PhD student at WashU in St. Louis. And Indigo Rose, who was my lab manager, who's now actually a graduate student at UCSF in Martin Campman's lab. But what they did is they took human iPSC-derived astrocytes, they treated with the same cytokines that we're using our rodent cells, and were able to fully recapitulate the transcriptomic changes that was occurring in this subtype of astrocytes. But importantly, we then had a cell-based assay that we could actually run our functional testing. And what we found was that these reactive astrocytes lose the same functions that they did in rodents, no longer able to produce synapses, no longer able to phagocytose debris and synapses during pruning. But importantly, for this particular subtype, they were releasing a molecule that was specifically toxic to neurons and not to other cells in the CNS. So we became progressively interested in this sort of phenomenon, this subtype. And so we decided to actually investigate this a little bit further using a number of different in vivo models of different neurodegenerative diseases, starting from acute injuries to the optic nerve, so looking at axotomy response, increasing that to longer and longer chronic neurodegenerative disease models to see if these astrocytes still played a major role. And so very, very briefly, I'll highlight two really interesting discoveries that were found, and we published these both last year. So in this particular report, paper in Cell Reports, Kevin again, who was the first author, showed that if we stop the formation of these neurotoxic reactive astrocytes in a bead occlusion model of glaucoma, we could actually stop the neurons from dying, but most importantly, these neurons retain some electrophysiological function. It wasn't the same as what they normally had in the control setting, but it was remarkably more than we anticipated there being. And we actually used this or interpreted this as evidence that it was actually worthwhile us investigating further how to create a therapeutic to stop the formation of these reactive astrocytes. And also spearheaded by Kevin, he was very busy in a paper published in Nature Communications last year. He actually showed if we slow down the generation even further, because the glaucoma model takes a month or so to manifest, we slowed it down even further using this SOD1 G93A high copper mutant mouse model of amyotrophic lateral sclerosis, which actually leads to progressive degeneration of motor neurons and death of the animals within 3 to 4 months. We could see that if he stopped the formation of these cells, he could increase the lifespan of these mice by about 50%, which is an incredibly large increase in lifespan in this incredibly progressive and aggressive neurodegenerative disease. So what we then had is an ability to study this astrocyte reactive response. We knew what the initiators were. It was immune cells in the central nervous system with microglia. We knew the cytokines that were inducing it. We knew that these neurotoxic astrocytes were releasing a molecule that was killing neurons. And we knew that if we stopped their formation, we could actually preserve the health and function of neurons. And so there's ongoing studies in the lab looking at how we can actually perturb that function at the earliest stages of disease initiation and progression. But what we were really interested in was exactly how heterogeneous this response was. How many different subtypes of reactive astrocytes exist within any one particular brain region at any one particular time in any one particular disease? And so Philip Haas in the lab decided to actually investigate this even further. And so here's some work that Philip and Mike O'Day have been doing, is actually looking at some of the largest single cell astrocyte data sets that have been generated to date. In this particular one I'm showing you here, we have 80,000 astrocytes, and this was published a few weeks ago in Nature Neuroscience. And what Philip was able to do is actually study the astrocytic response to inflammation in both male and female animals to try and determine if there are any sex-specific differences in this response. And we know that all of these cells that we're looking at in this 3D UMAP here that Mike generated are actually astrocytes because they express all of these canonical astrocyte markers. They're not expressing other cell type markers. And we also know, of course, the benefit or the strength of single cell sequencing is that there are lots of different subtypes of these reactive astrocytes express very specific differentially expressed genes that are specific to those subtypes. And so we're slowly going through the catalog now and determining which one of these subtypes may have putative important functions, may have important strategic positions in the brain, and we're investigating them further. And so the one I want to highlight very briefly today is this cluster down here, which is indicative of astrocytes that are responding to interferons. And this has the non-enthusiastic name of cluster 8. We colloquially in the lab call these astrocytes doomers, because in our minds, the names are not that important. It's the functions that are the most important for us. And this cluster of doom astrocytes was actually the only reactive astrocyte phenotype which was largely constrained to the inflamed brain. We never saw these cells in the non-inflamed brain, or very rarely. So they had changed their transcriptome so robustly that we couldn't actually determine where they had come from. What we could determine, though, is that these astrocytes are interferon responsive. They express a lot of interferon responsive genes like IGTP, TAC1, and a whole host of viral response genes. And with these differentially expressed genes, Philip was able to ask where they existed in the brain. And so just to show you very briefly, he found that they exist in the outer layer of the cortex, layer 1, underneath the meningeal surface. And he's shown this here by in situ for IGTP, this doomer marker, which is co-localized with an astrocyte marker, GLAST or SLC1A3. And he also importantly showed that these were not actually vascular cells that were expressing this marker, because you can see that they track along these penetrating vessels, but they were indeed juxtavascular astrocytes, because they didn't co-localize with endothelial cell markers like Claudin-5. So this is a nice visualization of an individual marker, but of course we want to be able to expand this and say, well, are all of the markers expressed in those astrocytes here that we identified, or that Philip identified, I should say, in his single cell sequencing data? And of course, what we can do now with the advent of amazing tools like spatial transcriptomics is we can actually do that experiment. So Philip did this, realized very quickly, of course, that the resolution of spatial transcriptomics is nowhere near what you would need it to be to look at cell type specific or individual cell responses. So this is actually some 10x Visium imaging of a saline injected brain and a lipopolysaccharide injected brain. This mounts a systemic inflammatory response. And we could argue that there are more dots on the bottom brain than the top brain, but the resolution of these dots is so large that we actually can't look at individual cells. And so what Philip did is, rather than looking at a single marker, he actually amassed all of the differentially expressed genes from each individual cluster, sometimes between 50 and 250 genes. And then he can use this module to probe into the brain and say, where do all of those genes get expressed together? And when he does this, lo and behold, he sees what he saw with his in situ hybridization experiments. There is expression or high module score on the surface of the brain where these cells exist, but he also saw that there was a hot spot around the ventricular system. Two very strategic positions in the brain where any astrocyte can interact with both the central nervous system immune cells, the microglia, but also the peripheral immune cells that will be coming through the bloodstream. So expanding on this, Michael Day then took that analysis and pushed it even further. We like to laugh in the lab that he threw math at it, and he took advantage of a Bayesian analysis next nearest neighbor analysis toolset that was published by Ed Zhao in Nature Biotechnology earlier this year, in which he's able to actually increase the resolution of the Visium image, plus combine that with Philip's module probing approach. And so when he does this, what we can see is that, of course, in the physiological brain, there are none of these doomers present, but in the inflamed brain, again, we see them on the brain surface and around the ventricular system. But just to show you that this is not the signal we get for all of these clusters, we can see that other clusters of reactive astrocytes exist largely subcortically, or may not exist largely in the cortex in the unaffected brain, but of course upregulate in the cortex following inflammation. And important for the study of Alzheimer's disease, there are astrocyte populations that exist largely in the hippocampus, but following inflammation, we can see there's an increase in the number of these cells tracking along this foramen that's under the hippocampus and then surrounding the ventricular system. And then extending this even further, Mike actually took advantage, took advantage of some human single nuclear sequencing we had been doing to then do some integration studies. So the first step of this was done by Jessica Sadik, in addition to collaborating with Philip. And the first step was to integrate it with mouse Alzheimer's disease model data sets that were available. So here she took a data set from Mikhail Schwartz's lab and one from Marco Colonna's lab, both of which were high quality data sets, but extremely underpowered because of the number of astrocytes they were able to capture. And I won't run through this dot plot completely, but it's just suffice to say there's a lot of genes along the bottom here which are expressed by doomers. If we highlight just a few of these in this particular doomer cluster and we look at these violin plots, we can see that they're largely constricted to the Alzheimer's disease models. And we can zoom in on one of these and just show you, here's that original marker, IgGP, of which Philip did this in situ on. We can see they're not expressed by the glutamate control mice, but it is enriched in the 5X FAD mouse model of Alzheimer's disease at 7 months, but actually they're not present in this mouse model at 15 months. And so we interpreted this as meaning that these doomers, given their strategic location, probably have a really important role in the earlier stages of the disease, either in staving off peripheral inflammation or perhaps releasing cytokines that can then recruit additional peripheral immune cells that may otherwise exacerbate the disease. And there is some evidence this could be really important. We look in some staining from human postmortem tissue. This was done 2 decades ago. We can see that another marker of these doomers, the cytokine CXCL10 or IP10, is lowly abundant in the non-symptomatic aged brain, but there is a gross increase in the amount of this marker in individual astrocytes, and also in the number of astrocytes that are specific to this marker. And then, like I said, my slide deck has changed to what I thought it was, but that's okay. We know that these astrocytes are responding to inflammation in different ways in both rodent and human iPSCs. We also know that the response is revolutionarily conserved across these species, but also conserved across different disease models as well. And why we were able to show in human diseases that this was present in a whole host of different diseases like Alzheimer's, Parkinson's disease, Huntington's disease, multiple sclerosis, etc. We're also able to show that this can have a real effect when we stop their formation in mouse models of glaucoma and also in mouse models of ALS. We also know that neurons can be saved from electrophysiological function, which is really important because it gives us a premise to actually go in and target astrocytes in the context of these neurodegenerative diseases. And we also know that because we're generating all these in vitro models to recapitulate the transcriptomic profiles we see in vivo, we can actually use this to study the functions and actually understand novel molecules that are being released by these astrocytes. And that's exactly what Kevin did to discover the fact that these astrocytes release toxic lipids. So I guess the question then is, is there more to life than reactive astrocytes? I talked a lot about this, and I said I wanted to talk about life a little bit, and there is. And so the last few minutes, I want to go through that a little bit. Obviously, you know that I have this affection for movies from the 80s. That's a cause of my upbringing. I also roast my own coffee. So the lab drinks through several pounds of beans every week. So I spend my weekend importing beans and roasting beans. And I just have to put this here because Jeff Goldblum's great, and how can you not love him? So you should go watch Jurassic Park. It's one of the greatest movies ever. But of course, apart from silly things that are really important, there's a lot of other really important things in life, and particularly in science life. And ultimately, what I want to spend the rest of the time is talking about mentor support. So you can have, as I said, you can have very helpful mentors and you can have very harmful mentors. And when I was an undergraduate, I wanted to get some experience in a lab, and I went to every lab in my department asking them if I could work in the lab over the summer. And they all told me I was too stupid to work in the lab. And they all collectively then told me when I was applying for graduate school that I was not smart enough to do a PhD. And I didn't know at the time because I just went to all the people who were teaching me because they were the only thing I knew about science. No one in my family had gone to college before, and I really didn't know what I was doing. But I did ultimately find exceptional mentors. I joined the lab of Katarzyna Dzingalewska and Norman Saunders, both immigrants in Australia. Kate is Polish, Norman was British, and they really took me under my wing and fostered my inquisitiveness in science. And so they taught me 3 really important things that I'm now trying to impart onto trainees in my lab and trainees in my institution. And the first is that you have to help people find what their passion is. And this sometimes takes a lot of time, but if you find out what people's passion is and you find out what they have a technical aptitude for, they're going to enjoy the science a lot more. For 3 years of my PhD, I did histology. And that doesn't sound particularly interesting, but I was really good at it. And so I was able to generate high quality results, which meant that I was in a much better mental state going through graduate school before I moved on to do a lot of in vivo physiological experiments. It's also really important, they taught me, that if you're going to work on disease, you have to meet a patient. It's very, very difficult to do translational research if you've never spoken to a patient about what ails them. often what we in a laboratory think is what we should be doing for patients may not actually be the thing that provides the most support to that patient. And so it's a rule in my lab that if you are working on a disease, you talk to a patient or carers or patient advocacy group, and we do a lot of outreach to make sure that we keep the patients themselves and the community abreast of what we're doing, and also take on board their suggestions and their insights into the disease that we don't have as non-patients. And the other one is that you just have to humanize the experience. We're not robots. We're not here data generating machines. We're people, and life happens. You know, people have children, people have mental illness, people have difficulties. They have things that need to get taken care of in an immediate sense. And you need to support your people to ensure that they're actually able to be the best versions of themselves outside the lab, so they can also be the best versions of themselves inside the lab. And there's a lot of examples I've had with trainees who've come to me worried about these life experiences because their PIs are not being particularly supportive. So following graduate school, I changed fields, I changed countries, and I was very, very fortunate to work with this absolutely exemplary, wonderful human being, Ben Barres. If you haven't read his autobiography, I highly recommend it. It's the autobiography of the Translational Scientists. It's available to purchase, but you can also get it for free on the SFN website, Society for Neuroscience. They have an amazing selection of autobiographies from a large number of exceptional neuroscientists. But Ben, of course, was a very outspoken advocate of everyone who was underrepresented, regardless of what group you came from. And I already had this mindset, and so it was just exemplified by being in his lab for 7 years. And he wrote very openly about this in 3 papers that I recommend everyone reads before they join a lab, at least 3. The first is where he really speaks out and talks about how you have to treat people equally. You know, if you see something happening, you have to say something. And it sometimes feels like our voice is very small and we don't have much ability to be an advocate. You can speak out in your friend group or in your laboratory or in your graduate program or in your institution. As you get more confidence, you can speak out more broadly at meetings and things. So you really need to take action to make sure that you support those with a lesser voice than your own. The other thing Ben wrote a lot about was that you have to choose very wisely the group that you're going to join. Either the laboratory you're joining, the graduate program, or ultimately the company that you work for, the institution you start your lab in. Because good mentorship and having good mentors is completely pivotal to your success. Your science will be good. If you're a good scientist, it doesn't matter what you work on, it will be good. But if you are not supported, it's very, very difficult to get a leg up. And it's really pivotal for the success of your science to have someone there who is your champion. And so it's really important that you choose carefully, talking to people who are in the lab now or talking to people who have left the lab and just seeing if they have moved on to positions that you yourself would like to get and asking if they felt supported in that role. And the third thing Ben wrote about very shortly before his death was really the fact that it was written mostly at mentors, not at the mentees, but essentially says that you have no right to be supported if you are not supporting your people. We shouldn't be giving you funds. We shouldn't be giving you labs. We shouldn't be allowing you to take on students and postdocs if all you are doing is using them to generate data to sort of continue to launch your egotistical ideas. So I urge you to choose a lab that provides support. Don't choose the lab that you think is doing the most flashy science. Often those things go together. So I'm not here to say that flashy labs are bad, but the support is the most important thing. So this is the last piece I want to finish on. This is my career trajectory. I was born in Australia in the early 80s. I moved on the West Coast. I moved to the East Coast after high school to do my undergraduate study and my PhD. I then moved to Stanford. I then moved to NYU. Very, very simple trajectory. But I think what is missing sometimes is people realize that all trajectories are not simple and everyone has a different story that you may not know about. So it's always nice just to ask people to tell you, how did you get where you are? Because you can learn a lot about their sort of ethics and things like this from asking that. So I was born in a town of less than 4,000 people. No one in my family had gone to college, let alone having anyone who had done any science. I had no idea what a mentor was. I'm sure I'd read the word, but who knows, right? It's a small farming town. I then actually went to college. I was really interested in doing biochemistry. I ended up actually getting mugged in my first year. I had brain surgery. I quit school. I couldn't concentrate. But I was taken by the fact that I saw my own brain on the operating table and was really interested in then studying neuroscience. But as I told you, I was told I was not smart enough to do a PhD. So I like to tell people I had really terrible mentors. They just were not good at inducing any excitement in the trainings they had. They were just there to boost their egos and generate data. I then actually switched fields. I actually ended up doing my PhD in pharmacology of all things, but I worked on the blood-brain barrier, but I had exceptional mentors. But I totally acknowledge that even with exceptional mentors, life is hard. You know, I nearly quit my PhD. I started a side business. I was working as a wedding planner. I left Australia. I traveled a lot. It is mentally draining to go through these experiences. And the thing that pulled me through was actually having a really good cohort. And even just this morning, I was texting with one of my graduate student cohorts, just talking about the fact that I was presenting today. And she said, you have to make sure you point out that it was hard for you. It was hard for all of us. But if you have friends, you can get through this. And unfortunately, due to the bad mentors that she had, she left active science, but now is actually running clinical trials in a major hospital and loving her life. So a PhD can get you in lots of different places. My postdoc seemed like it was a great thing, moved to a new field. But again, in the first year of my postdoc, I was hit by a car, broke nearly all the bones in my body, and spent 4 years doing rehab where my hands didn't work, but it made me focus a lot more on the patient side of things. What is it patients would actually need to actually improve their lives on the daily, as opposed to what would they need in 10 years' time if you could develop new therapeutics? And we always try and think about these two things in the lab. Of course, as I mentioned before, unfortunately, my advisor was diagnosed with cancer, and that was an incredibly difficult time, but it did have some tiny silver lining in that I got to be his proxy and travel a lot giving presentations. So I got to meet a lot of exceptional people, and I think that was one of the reasons I ended up with the position I have now. But ultimately, I had an amazing mentor who really jettisoned my career. And now I'm at a school that I specifically chose because of the mentor support. And it's really, really exciting to be in this role because this is what I really wanted to do. I really enjoy the unknown that we are discovering. I really enjoy seeing all the people who are working. I really enjoy working with trainees and sort of making them the best versions that they can be. But you also get to do other cool things. I've started a biotech company. I'm looking at starting other companies. And I get to do a lot more work making sure that research is fair and equitable for those other people who may not be as lucky to have a loud voice that I now have with the platform I'm on. But it's also terrifying because I am now the mentor and looking back and making sure that I'm able to look after those people in my lab. So last slide, and this is just to say, you don't have to wait till you're in charge to make a difference. You know, I mentioned that you can be very active now and give a voice to those who don't have it. The goal is to normalize those underrepresented minorities, normalize their voice and normalize their concerns. And it doesn't matter if it's your friend group or your lab or a larger institution. You also can get involved in a lot of things that you may not realize that can actually help to increase diversity, but also increase the feeling of belonging that these individualized groups may have. So you can organize meetings or ask your mentors to invite seminar series to get a diverse set of speakers in to engage with those visiting scholars and make sure that you don't have the same speakers coming in all the time. And this sounds a little bit sort of contrived, but you are actually the future of research. You know, you get the choice to make this future the future that you want to live in. And you don't have to listen to what I say, because what I say in 10 or 15 years' time may be old hat. So you get to create that community that you want to live in. And you should do that. And then you should teach me what I've done wrong. And then ultimately, when you get there, you have to look back and bring those other people with you. Don't climb the ladder and bring it up after you. Climb the ladder and build a staircase to help those who can't get there as easily as others may be able to. So that's all I wanted to say. It was a rambling story. I have already thanked all of the amazing people that did the work actively on each slide. But these are their names again. These are their faces. These are their contact details. These are the papers I talked about. And we always try and make our data very open and available, so you can see it all here. And finally, I'd like to give an enormous thanks to all of our supporters, either by providing financial support, but also in particular the patients and families who've donated their tissue that enabled us to do all the amazing work that everyone in my lab does. So I know that I went longer than I probably should have. I do apologize. I've become that old professor now. But if we do have time for questions, I would love to address them. But please feel free to tweet at me. We have a handle now, or drop me an email. I'd love to chat some more. So thank you very, very much.
Thank you, Shane. That was a really nice talk. I really enjoyed it. And it's always really, really refreshing to hear from a PI who has your stance on supporting early career researchers and making sure you're paying things forward rather than, as you say, pulling the ladder up behind you. So that's really, really nice to hear. If you don't mind, I'd like to start with just a quick question, and we do have one in the chat as well. So the one in the chat is a science one, so I'm going to start with one on the career side of it. You mentioned how you had really terrible mentors as an undergraduate, and you had to seek out actively good mentors for your PhD and your postdoc, and it sounds like you did that very successfully. How did you go about finding those mentors? How did you know that they were going to be good mentors, and how did you make those connections? Did you do cold emails? Do you have any advice on how to approach those conversations?
Yeah, that's a good question. I think the first time I did it, when I worked with Cátia and Norman, was complete serendipity. I just happened across them at the end of the corridor whilst being despondent about not having any success elsewhere. And they were just so warm and welcoming. And there were people in the lab already, other trainees, postdocs and students, and they were having a good time, and they were actively talking about the science. And I was like, this is the community I want to be in. And I was very, very fortunate they took a chance on me. So that was complete serendipity. The way you do a PhD in Australia is very different. It's a bit more like the British system, where you take a form to a professor and say, I would like to do a PhD with you. So it's a little different in the US system, where you go through interviews and you enter a program, but you should be asking those questions as well. You should always be talking to the trainees in the labs. When I joined Ben's lab, and I acknowledge this has been really hard in the last 2 years, but the way I actually realized that I wanted to be in his lab is that I met him at a meeting. We were sat next to each other in the front row at a Cold Spring Harbor meeting, and we were discussing the science that was going on. And I was, interjecting with questions and was really pressing the speakers about whether or not the interpretation of their results was correct. And Ben thought that I was thinking about science the way that he liked to think about science, and the way that people in his lab thought about science. So again, it was somewhat serendipitous, but after I chatted to this random guy next to me, Ben Barris, who was this, I read his work, I appreciated it. I looked at where people had gone in his lab. I wanted those positions, and I talked to those people who had left his lab and asked, what was it like? What training did you get? And it was the training that I wanted. So when I say, I don't want it to make it like victim shaming, but I want to make sure it's sort of upon you as a trainee to make sure the environment you're going into is the right environment for you. And different people require different mentor support. There are other exceptional mentors that would have been terrible mentors to me. But that was the sort of mentor support I needed. And I also fully acknowledge, as a mentor, I can't be everything to everyone. I can help my trainees in what it's like to be an immigrant in the US or a scientist, but I can't give them a lived experience of being a woman in science, or a person in color, or a member of the LGBTQI community as well. So I think you need to build a mentoring team that enables you to be the best version of yourself, and don't rely on a single person, because as I know all too well, sometimes that person can be gone, and so you want to have a lot of other people that you can turn to. That was a long answer to a short question. I apologize.
It was quite a long question as well, so thank you. That was really helpful. And so just briefly, we have a question from Joshua Hanley in the chat, who would like to know, do you have any plans for mass spec imaging similar to your spatial transcriptome analysis?
Oh, that's such a great question, Josh. I don't. I feel really bad that I can't give you a definitive answer that we're doing it. We are doing a lot of mass spec to look at the secreted molecules that astrocytes have in different reactive states. One of the reasons we're particularly interested in that is astrocytes tend to communicate by secretions, proteins, lipoparticles, exosomes, things like this. So this is the avenue we've gone down. But mass spec imaging sounds awesome. It's not something that we have the capacity to do at the moment. So I hope that someone on the call does that, and I read an amazing paper that you have done looking at this, because that would be awesome. If we can help and share the spatial to combine with that, please let me know. I'm happy to do that as well. That would be really exciting.
Great, thank you. So we'll move on, but thank you so much, Shane, for speaking. And are you happy if anyone has further questions for us to bounce those to you, or maybe they can get in touch with you on Twitter?
Absolutely. So long as they tweet at me with a picture of a gremlin, I'm very happy to answer.
Awesome. So we're going to move on to Dr. Charlie Arber. So Charlie, if you want to share your screen. Charlie is a senior research fellow funded by Alzheimer's Society at University College London, and he's going to talk about his work using stem cell models of dementia. So ready when you are, Charlie.
Thank you, Sarah, and thank you to Proteintech for the great opportunity to present some of the work. I feel it's a bit unfair to follow Shane, but anyway, I'll be talking to you today about how we're looking at familial Alzheimer's disease mutations in presenilin 1 and how they affect human stem cells in a manner of premature neurogenesis. So just as a quick outline, I'll first of all introduce some of our stem cell models of familial Alzheimer's disease, and then mention how we're using these to model adult hippocampal neurogenesis. Just as one slide of familial Alzheimer's disease background, so this is an autosomal dominant inherited form of dementia. It represents around 1% of all Alzheimer's disease, and it's caused by mutations in 3 different genes, namely the amyloid precursor protein shown here, and presenilin 1 and presenilin 2, which form the catalytic subunit of an enzyme called gamma secretase. The fact that APP and presenilins exist on the same molecular pathway to produce amyloid beta peptide led to the amyloid cascade hypothesis, which suggests that these mutations affect the production of amyloid beta, somehow leading to its aggregation and deposition in amyloid plaques, which somehow leads to intraneuronal tangles of tau protein, and again somehow leads to neurodegeneration later on in advanced Alzheimer's disease, shown here via a degenerating brain post-mortem. These 3 pathological hallmarks look almost identical in the early onset and the late onset forms of the disease. However, the mechanisms behind how these pathologies interact to lead to degeneration remain really poorly understood, and it's really my belief that this is absolutely crucial to understand if we're going to go on to tackle Alzheimer's disease, to understand the basic biology behind it. And with that in mind, there are many models and tools available to study neurodegeneration and Alzheimer's disease. Each of them, of course, has its own pros and cons, but these models can range from looking at individual proteins in a tube to genetics and brain scans during life and post-mortem tissue and complex animals. But there's really one thing missing from this jigsaw until recently, and that was the ability to study human brain cells in a dish. And that's for obvious reasons, that you can't go around biopsying people's brains willy-nilly, as it were. And that's really where stem cell technology comes in. And here at UCL in London, we're very lucky to work really closely with our clinical colleagues and some really motivated research participants with some of these gene mutations of interest, who donate their skin, and we grow their fibroblasts into a resource that is readily shareable worldwide. And we can take these fibroblasts, reprogram them to iPSCs. I think we'll hear a lot about iPSCs today. We do this via non-integrating methods, such as episomal reprogramming. And then these can generate any cell type in the body. And of course, we're interested in cortical neurons as the cell type affected by disease, by dementia. We follow these developmental paradigms. So this thing takes around 100 days in vitro. And as well as doing this in 2-dimensional culture systems, we generate cerebral organoids in 3D as well, again, taking around 100 days. But the benefit of this is that, as you can see, this is a bit more of a permissive environment, and the cells are able to organize themselves into these distinct layers representing the progenitors and the neurons, which is a little bit more akin to brain development, if you like. Once we've made these neurons, we can show that they're functional. So this is a 12-well plate with a multi-electrode array. So each well has 96 recording electrodes. And hopefully what you can see is that there's neuronal activity spontaneously in all of these wells, and even some networking and bursting you might see in this well, if you keep an eye on that well. There. Okay, so what's important to note is that obviously these cells come from the research participant with the gene of interest. And so for the first time, we can study brain cells in a dish from human subjects without overexpressing a gene of interest. So working with familial Alzheimer's disease, the first thing we wanted to look at was how amyloid beta is processed and how it's released into the media from our stem cell derived neurons. And just to mention, A beta doesn't exist in one form, but it's progressively trimmed like Pac-Man down smaller and smaller species of amyloid beta to 40 amino acids in length and 38. A beta 40 is the most abundant species, and these slightly longer forms, such as amyloid beta 42, are slightly more hydrophobic and aggregation prone and thought to be more toxic. So when we looked in our panel of stem cell derived neurons, we've got our controls here in gray, and we've got up to 9 or more stem cell lines now with different mutations in either APP or across the presenilin peptide. What you can see is that all of these mutations increase the relative production of this toxic species, as it were, relative to the abundant amyloid beta 40 species. You can start to see some of the differences between mutations, and we're really interested in the clinical heterogeneity and some of the differences between mutations, such as amyloid beta 43, which I don't have time to talk about today. But some of these differences are really interesting in how patients might present to the clinics and some of these differences. So how good is our model? So what we've done here is we've compared the ratios relative to controls for all of these mutations in our stem cell derived neuron supernatants. And we've taken patient plasma. So these are matched mutations, and the red dots are actually the same research participants. And we're able to see really high concordance between our in vitro model to what's really going on in patients. And I think this is a really good way of showing that we can accurately model exactly the effects of some of these mutations on amyloid beta processing. Some of this work was done very closely with a PhD student in the lab, Jamie Toombs, who I'd just like to acknowledge at this point, before moving on to say, we've taken this model forward to start to try to understand some stem cell biology in some of these models. So with that in mind, I'll talk about human stem cell modeling of adult hippocampal neurogenesis. So we're interested in adult hippocampal neurogenesis because this is a process whereby stem cells in the brain persist through life and generate new neurons right through aging. This process has been shown to be altered in rodent models of Alzheimer's disease and very closely linked to cognitive decline. However, in humans, of course, this is impossible to study during life, and postmortem tissue only shows the very end point of adult hippocampal neurogenesis. And there's only been a small number of studies, and these have had quite varied results. So we saw this as a bit of an opportunity. And we were quite excited to learn that the neurogenic programs shown in adult hippocampal neurogenesis are really highly conserved with developmental neurogenesis. Here are a couple of papers suggesting this. And what I mean by that is that the programs for neurogenesis during fetal development are used again in aging as these adult hippocampal neurons are generated through life. So we wanted to ask, can our in vitro neurogenesis model be a surrogate to study what happens in familial Alzheimer's disease and adult hippocampal neurogenesis? And this would otherwise be impossible to study during life in humans. Just as a quick aside, I'll talk about Notch signaling. So Notch signaling is crucial for all stem cell populations in development and aging. And this is another substrate for presenilin. So presenilin and gamma secretase, as well as cleaving APP, cleave around 100 different substrates. And gamma secretase and presenilin is responsible for the rate limiting cleavage step of Notch signaling. And so you might suggest that this is crucial for stem cell maintenance. Indeed, again, going back a couple of decades, mutations in presenilin-1 have been shown to impair Notch signaling. And so this really led us to the hypothesis that mutations in presenilin-1 might alter neurogenesis via impaired Notch signaling. So I mentioned that neurogenesis programs are similar between development and adult hippocampal neurogenesis. And just to show this, I've got some data here that we've looked at from the Allen Brain Atlas in BrainSpan and single cell RNA sequencing. And I'll just talk you through this. So when we look at human cortical development, you can see the different layers of the developing cortex shown here. And what you can see quite nicely is that presenilin 1 and the gamma secretase components are really enriched in the progenitor layers and similar to Notch1, you can see here. This is in contrast to APP, which is enriched in the postmitotic neurons in the human developing cortex. And we can go on to our cerebral organoid models to show, to distinguish the neural precursors shown here in the green colors and the postmitotic neurons. And again, as you might expect, Notch is very highly expressed in the progenitor layers. APP is expressed in the neural layers, and presenilin 1 exists in the same space and time as Notch signaling in these progenitor layers. Similar to that, when we look at, we reanalyze this single cell RNA sequencing database data set, and what we were able to see is that the neural stem cells cluster here compared to the immature precursor cells and the newborn neurons. And similar to what we've shown here, Notch and presenilin expression is highly enriched in the stem cells, whereas APP becomes more highly expressed as the cells mature. And so we can suggest that the neurogenesis programs are similar, and that Notch and presenilin 1 are spatiotemporally enriched together, so they're interacting. So what are the effects of presenilin 1 mutations on neurogenesis? So for this, first of all, we turn to our cerebral organoid model, and at around day 45, we quantified the relative proportion of the progenitor cells shown here in magenta compared to the postmitotic terminally differentiated neurons shown here in green. When we quantify the relative proportions, hopefully you can start to see that when there are mutations in presenilin 1, we start to see a relatively lower proportion of contribution from the progenitors and a relatively higher proportion of postmitotic neurons. And this suggests that we're seeing this earlier differentiation phenotype. We wanted to take this further in a parallel orthogonal system, so we turn to our 2D system, and hopefully even by morphologically, you can see that some of these lines with presenilin 1 mutations have this very early neuronal phenotype shown in this image here. We can quantify this by high content imaging for neuronal markers, and you can see a trend to increased neuronal content in our presenilin mutations in salmon color and lime green compared to our controls. And we can confirm this by qPCR on the population level, where neuronal tubulin is significantly higher in our presenilin mutant lines compared to controls. Importantly, at the same time, Notch readout genes such as FABP7 are significantly reduced, and this is suggestive that there's less active Notch signaling going on in these cultures with presenilin 1 mutations. And we can confirm that at the protein level, where we can see more neuronal tubulin and less active Notch signaling, especially in these quite severe mutation lines here. So all in all, this suggests that we're seeing this premature neurogenesis phenotype that results from mutations in presenilin 1, and this is at least in part due to reduced Notch signaling. Finally, we turn to postmortem tissue, and no one had actually looked in gene-confirmed postmortem tissue from familial Alzheimer's disease before, and we were lucky to have access to a small number of postmortem samples, 5 controls and 7 familial AD cases with gene-confirmed cases. And to quantify neurogenesis postmortem, we looked at the number of newborn neurons marked by TUJ1 crossing the granule layer of the dentate gyrus. Hopefully what you can see is there are small numbers, but we can start to see deficits when we have mutations in presenilin 1 compared to our controls. Of course, the tissue is incredibly rare, and these findings aren't significant, but I'll draw your attention to some interesting observations, such as this particular mutation seems to behave like the controls. And when we look at in vitro biochemical data, the cleavage of Notch biochemically seems to behave very much like controls when we have this mutation in vitro biochemistry from these published papers. Going back to the work on the previous slide, again, you can see that this mutation seems to behave more like controls, So I know the numbers are small, but there's a chance that we're starting to see the clinical heterogeneity that I mentioned sort of coming through again, these mutation-specific effects. But all in all, it looks like these results are supportive of this premature aging effect of familial Alzheimer's disease on neurogenesis. So just to sum up, we've been able to model an otherwise inaccessible part of human biology by looking at stem cell neurogenesis in vitro. And hopefully I've convinced you that mutations in pre-SNL1 lead to premature neurogenesis, and this is at least in part due to reduced Notch signaling, potentially leading to this premature aging effect. And just as my last slide, I'd like to sort of shout out to the stem cell model, because by working in the cell type of interest and without using overexpression models, we've been able to really compare some of these phenotypes between different mutations. And I mentioned sort of some of these patient heterogeneity, so some of these more severe phenotypes that you see in different lines. We can take this further to try to understand exactly how these mutations are working, such as via CRISPR, and you can have 0, 1, or 2 copies of the mutation, which will give you information about how the mutation is working, such as gain of function versus loss of function. And finally, you can start to build up the complexity of these models by adding in stem cell-derived astrocytes and microglia, like we're doing in the lab now, and some of these 3D culture systems that will help us to model the brain a bit better. And with that, I'd like to thank everyone in the Ray Lab, especially Chris Lovejoy for his help with the organoid work, and Jamie Toombs for his help with the amyloid beta, and especially to Proteintech for making today possible. It's been a privilege, so thanks very much, and I'll take any questions.
Thanks, Charlie. That was another really nice talk. And as always with the iPSC work and organoids, we also get to see really pretty pictures, which is always very cool. So if anyone has any questions for Charlie, type them in the Q&A now and I'll read them out. I wanted to start just by asking some questions about your 3D organoid model, if that's okay, because it's not something I've worked with before, so it's quite exciting. I wondered, so you said that the cells are forming layers within the organoid. How do they resemble structurally the layers that you actually see in the cortex of the human brain?
So I think the organoids suffer from getting too large to have effective transfer of nutrients between sort of the deeper layers. So they start off really well, but to get the actual layers of the cortex, it turns out to be quite difficult. And some of the newer models, such as the alicos, where you can take slices of the organoids and then grow them in 2D, those ones actually develop into these beautiful layers, which are more akin to the developing cortex. So I think it's not quite there yet because of the problems with perfusion and cell death in the center of the organoids. But at the moment, they're more of a bit of a jumble as they get older, I think. Yeah,
I mean, it's very cool either way, but I was just being curious. I've got a question in the chat from Shane. He says, great work, Charlie, capital great. Do you see a benefit in generating organoids from a combination of mutant and isotype control or wild type cells to look at the effects of individual cell types within the mutation, or even generating mutant and wild type connectoids?
100%. I think that's so exciting, and to pick out the differences, the different contribution of mutant astrocytes, for example, in wild type neuronal cultures, I think would be really important going forward, and that's some of the work we're doing now. Personally, I prefer 2D cultures because you've got much more control over the culture. The cells are much more homogeneous. I've been growing 2D, growing cerebral organoids. There's always this joke, they're great for some things, but they're so heterogeneous. You end up with toenails in there and kneecaps, and not quite, but at the moment, you need single cell sequencing to really pick out some of the phenotypes in there.
Okay, cool. We've got a question from someone anonymous in the chat. Say, thank you, Dr. Arber, for the great talk. Can you do electrophysiology in the iPSC model? And what's the advantage and disadvantage of this method compared to in vivo models?
You can definitely do electrophysiology. I took out a slide because I don't really understand electrophysiology, but the stem cell derived neurons do behave like mature neurons, especially when you culture them with astrocytes. So again, the idea of support. And I think, you know, the benefits of using human stem cells is that we've got human cells. So when you think of something like tau splicing, I haven't talked about tau today, but mouse cells and human cells behave very differently. And one example of that is how they might splice disease-associated proteins like tau. So this is crucial for frontotemporal dementia and Alzheimer's disease.
Great, thank you very much. We do have a couple more questions, but in the interest of time, I'm afraid we won't be able to answer them, but they're in the chat, so maybe you could have a look, Charlie, during the next talk, or maybe you could discuss it afterwards. But thank you very much. And we're going to move on now to our next speaker, which is Anushka Bhargava. Do you want to share your screen, Anushka? If you're ready. So Anushka is a PhD student at the Sheffield Institute for Translational Neuroscience here in the UK. And she's going to talk to us today about some potential novel therapeutic targets in ALS. So ready when you are.
Thank you, Sarah, for that introduction. So good morning or afternoon, everyone. I'm Anushka Bhargava, and I'm a third-year PhD student at the Sheffield Institute for Translational Neuroscience. And today I'll be talking to you about my PhD project, which focuses on identifying erfaptin-2 as a potential therapeutic target for ALS using iPSC-derived motor neurons and zebrafish as models for ALS. So just to give you all a brief background on the disease, ALS is a rare adult onset neurodegenerative disorder that is characterized by an accelerated loss of motor neurons, which are basically responsible for controlling voluntary muscles. And the average age of survival after the first sign of symptoms is about 3 to 5 years. And currently for the disease itself, there's no known cure. However, disease management options include drugs like riluzole and edaravone. And though these pharmacological options are available, supportive care still remains the foundation for ALS management. So the pathogenesis of ALS is highly varied and multifactorial, and the exact mechanisms causing motor neuron death are unknown, but it is believed to be a complex interplay between the various identified factors here. And as you can see, that these factors are influenced by many genetic mutations, which makes ALS a genetically heterogeneous disorder. While the exact cause of motor neuron loss is varied, a key pathological feature is the presence of protein aggregates in neurons. So these protein aggregates are insoluble intracellular cytoplasmic inclusions, which comprise of misfolded proteins, and these may form when the degradation capacity of a cell is exceeded or due to defects in the protein degradation pathways. And the protein composition of these aggregates in neurons varies according to the disease background in ALS, but several common constituents of these aggregates have been identified over the years. And the first one to be identified as a constituent within the aggregates was the SOD1 protein, which is also the most studied protein in ALS. And another major constituent in more than 90% of ALS cases is TDP43, while the others include the FUS, the P62, and the ubiquinol proteins. And recently, 2 independent studies have shown that a novel protein called erfaptin-2 co-localizes within these aggregates in neurons and may be of therapeutic potential. So just to give you an idea of the function of erfaptin-2, it is ubiquitously expressed with high expression seen in several tissues, including the brain and skeletal muscles. And while the precise function of erfaptin-2 itself is unknown currently, its structure and protein-to-protein interactions can indicate possible functions. So there is a domain in its structure, which indicates that it may be involved in membrane binding, sensing membrane curvature, and inducing membrane tubulation, as well as it may play a vital role in the biogenesis of secretory storage granules. The protein is also known as partner of RAC1, which indicates that it may interact with small GTPases that are enzymes that function as molecular switches and can regulate a various amount of cellular processes within the cell. And studies into erfaptin-2 in neurodegeneration have added further to its proposed functions, and it has been shown to be involved in regulating protein aggregation and exerting neuroprotective effects. So previously, a study looked at erfaptin-2 in Huntington's disease and reported that while the full-length erfaptin-2 promoted aggregation, expressing only its C-terminus inhibited this, but this was not the case in the presence of a proteasome inhibitor, which hinted that the protein itself may be working to affecting the proteasome activity. And so they also showed that with an increase in the concentration of erfaptin-2, there was a decrease in proteasome activity, but expressing the C-terminus of the protein rescued proteasomal activity, thereby exerting its neuroprotective effects. Another study showed that the neuroprotective effects of the full-length erfaptin-2 depended on its phosphorylation status and showed that its phosphorylation promoted survival and decreased aggregation. And this phosphorylation of the protein was mediated by AKT, which is a pro-survival kinase. And although the two studies mentioned here describe different mechanisms by which erfaptin-2 is neuroprotective, it is noteworthy that the phosphorylation site is on the C-terminus of the protein. So the pathway by which erfaptin-2 may be therapeutic and neuroprotective may be the same. And since protein aggregation is a common feature in neurodegenerative disorders, therapeutic strategies can be transferable. And so previously, our lab looked into erfaptin-2 in ALS, and preliminary data showed that the C-terminus significantly enhanced survival in primary motor neuron cultures. So with these study and data in perspective, we hypothesized that erfaptin-2 may be a novel therapeutic target for ALS, targeting of which may ameliorate the disease phenotype. Specifically, we aim to investigate the expression and distribution of erfaptin-2 protein in healthy and diseased biosamples, which include the iPSC-derived motor neurons and human brain tissue sections, as well as assess whether the C-terminus itself can affect disease development in our in vitro and in vivo model for ALS. So we have used iPSC-derived motor neurons as our in vivo model, and the timeline here shows the established differentiation protocol that we use. So human-induced pluripotent stem cells go through neural induction and caudalization till day 12, at which stage they are neuron progenitor cells. And from here, with neuronal induction and maturation, they are differentiated into functional neurons at day 28 of the protocol. So using iPSC-derived motor neurons, we examined the endogenous levels of erfaptin-2 in ALS, and an increase in erfaptin-2 levels in ALS was observed. And to see the expression and distribution of erfaptin-2 in human brain tissue sections, cerebellum and frontal cortex sections from healthy controlled and sporadic ALS patients, as well as ALS patients carrying the specific C9 mutation, were stained for endogenous erfaptin-2. And here we can see that the ALS sections from patients with the C9 mutation were stained slightly darker in the cerebellum, especially in the Purkinje cells, as indicated by the arrows. And analysis of the sections showed that the intensity of the staining for endogenous erfaptin-2 was significantly higher in the cerebellum for ALS patients with the C9 mutations, as well as in both cases of the ALS patients in the frontal cortex sections. So with the C-terminus showing neuroprotective effects, a lentivirus expressing the C-terminal of erfaptin-2 was made for efficient delivery to motor neurons. And iPSC-derived motor neurons were transduced at different doses to select the optimal dose for subsequent experiments. And here we can see that the expression of the C-terminus was seen in all cell lines, and the expression was dose dependent. And using untreated cells for each line as a control, transduction at an MOI of 40 showed the highest transduction efficiency, while the cells still looked healthy 4 days post-transduction. And this dose of MOI 40 was chosen as the optimal dose for the lentivirus treatment for subsequent experiments. So our iPSC-derived motor neurons carry the C9 mutation, which is a hexanucleotide repeat expansion mutation in the C9 ORF72 gene. And this is the most common mutation in ALS. And the mutation itself is in the first intron of the gene. And the exact disease mechanism of this mutation remains unclear, but 3 mechanisms have been proposed. The first is that the repeat expansion could interfere with transcription processes, leading to a reduction in the C9 levels and hence a loss of function of the gene. Secondly, through bidirectional transcription, the repeat expansion might form secondary sense and antisense structures and nuclear foci, which can then sequester functionally important RNA binding proteins in the nucleus. And thirdly, through non-ATG repeat associated translation, toxic dipeptide repeats or DPRs can form, and they are generated from the sense and antisense strands. So an initial experiment conducted to look at DPR levels in iPSC-derived motor neurons showed that the DPRs were significantly increased in ALS, but when they were treated with the lentivirus expressing the C-terminus of refractin-2, there was a significant decrease in these levels, highlighting a possible therapeutic potential for the C-terminus of refractin-2. Several studies over the years have also indicated that ALS patients almost universally display the abnormal mislocalization of a protein called TDP43. And in normal circumstances, as we can see here, the TDP43 protein should originally reside in the nucleus of the neurons, but instead in disease, this is flushed out into the cytoplasm where it can then build up into aggregates. And this mislocalization can be seen in our in vitro model as shown here. However, when the ALS patients were treated with the lentivirus expressing the C-terminus of the disease, of refractin-2, we can see that there seems to be a potential rescue of this phenotype from what we can see in the ALS untreated cells. We also looked at cell viability in our in vitro model, and we can see here that the cells treated with the lentivirus with the C-terminus were significantly healthier over time than those that were untreated ALS cells. For in vivo analysis, we're using zebrafish as our model for ALS, and to establish stable lines expressing the C-terminus of the refractin-2 protein, the construct here was made, and this construct was then injected into zebrafish embryos at the 1-cell stage. And here we can see that the fish successfully expressed the transgene, as we can see in the fish 5 days post-injections. So those fish, the transgenic fish that were injected, were then grown to adulthood to be screened for positive founders. And of the 31 that we screened, we found 4 positive founders, of which 2 of them, highlighted here, lines 2 and line 4, have been established successfully. And here we can see that analysis of the line 2 shows a stable expression at 5 days post-fertilization and showed that the transgene was ubiquitously expressed as expected. The transgenic fish were then crossed to our zebrafish ALS model, and the fry was sorted into 3 populations depending on the phenotype. And these embryos shown here have been genotyped and sorted accordingly. And post this, we are hoping to assess the effects of the C-terminus on RNA foci, DPR levels, and motor performance in zebrafish. To summarize, ALS is a rare adult onset neurodegenerative disorder that cannot be cured or slowed significantly. And refractin-2 was found to co-localize within aggregates, and its C-terminus has been found to be neuroprotective. We have shown that the expression of refractin-2 is elevated in ALS, and this supports a hypothesis for targeting it in the disease. And from here, we aim to identify refractin-2 and in specific its C-terminus as a potential therapeutic target for ALS. Future work of my project would include further assessing whether the expression of the C-terminus can affect RNA foci or proteasome activity in our ALS C9 iPSC-derived motor neurons, in addition to those phenotypes mentioned today, as well as studying the effects of the C-terminus on disease development and motor performance in zebrafish. Finally, I would like to thank all my supervisors for their guidance throughout my project, and Dave, Cátia, and Toby for helping me and supporting me through my in vitro and in vivo analysis and experiments, as well as the entire aquarium staff and team at the Bateson Center. Thank you for listening, and I'd be happy to take any questions.
Brilliant, thank you very much. That was another really nice, clear talk and some really interesting data there. There's a question coming from James Quinn. He said, exciting work, Anushka. Do you know if it's possible to detect RFIP2 in the CSF or blood? It would be great to see if this was changed in ALS and could represent a potential biomarker of disease.
So that's an excellent question. This is something that we haven't looked into in my project, but this would be something that I would be willing to look into if I have more time in my PhD.
Yeah, there's always so much to do, right? But it does seem like a good maybe future avenue, I guess. I wanted to ask, so you mentioned at the start that the protein, the RFIP2 protein sort of co-localizes with a bunch of different types of protein aggregates in different diseases. Has anyone looked at that in C9? Does it aggregate with the GPRs?
So there's very limited data on F-aptin2 in neurodegeneration, and nobody has looked into it in ALS in specific. So the short answer to your question is no, but they have looked at it in Huntington's disease, and they have shown that it co-localizes with mutant Huntington aggregates. So they have looked at in Huntington's, but not ALS in specific.
Okay, cool. I think it's really cool that you're showing a reduced DPR burden as well, because that's something that's quite exciting at the moment. Do you have any ideas? Do you think that the mechanism of that is probably through the proteasome based on your previous data and through the C-terminal fragment specifically?
So like I said, that previous papers have pointed out that the C-terminus might be neuroprotective through affecting the proteasome activity. This is something that I'm planning to look in the next few months into in our ALS model as well. As for now, I cannot comment if there is a direct mechanism, but I do think it might be affecting the proteasome activity by which then the DPR levels are reduced in ALS C9 models.
And one more question from me, I promise, and then we'll move on. But I think that I actually just maybe missed this a little bit. I think you did mention it, but was it all of the DPRs where you saw a reduced burden, or was it different between the 5 different peptides?
So the ELISA that we conducted was the GP ELISA. So in specific, it was looking at the GP DPRs. We don't have an established ELISA for the other DPRs as yet, but this is something that we were looking into.
Maybe we should talk later, because I have individual DPR models that we could do some cell work. We'll talk later.
That'd be great.
But if there aren't any other questions from the audience, we'll move on. Thank you for a really nice talk, Anushka. And if anyone does think of additional questions, then of course, drop them in the chat and we'll make sure that you get to them. So the next speaker is Dr. Cátia Gomes. So if you want to share your screen for us, she's a postdoc at the University of Indiana in the States. And she's going to talk to us today about the role of astrocytes in retinal ganglion cell degeneration in glaucoma. So over to you.
Okay, thank you so much for the introduction. So yes, I'm Cátia, and I'm a postdoc in Dr. Meyer lab at Indiana University School of Medicine. So first of all, I would like to thank Proteintech for this amazing opportunity to be part of this panel and to present our research today. So I'm going to talk about the contribution of astrocytes to retinal ganglion cell degeneration in glaucoma using human pluripotent stem cells as models of disease. So as you all know, glaucoma is the most prevalent irreversible blinding disorder that affects around 70 million people worldwide. And this neurodegenerative disorder is characterized by the progressive degeneration and death of retinal ganglion cells, or RGCs. So while in physiological conditions, astrocytes contribute for the homeostasis and proper function of RGCs, in glaucoma, these glial cells become reactive and dysfunctional, so they lose their neurosupportive role, and they contribute for the degeneration and death of RGCs, leading to loss of vision or blindness. So although rodent models have been important models to understand developmental and neurodegenerative mechanisms, there are important molecular and cellular differences that have been observed between rodents and humans, and in particular, if we talk about RGCs and astrocytes. So trying to overcome these limitations, we can use human pluripotent stem cells. So we know that we can isolate somatic cells from patients. we can reprogram these cells to pluripotent stem cells and then differentiate in several cell types that then can be used for disease modeling or to do drug screening or to test other therapeutic strategies. So among all the risk factors that are associated with glaucoma, family history and genetic mutations are part of these important factors involved in the development of glaucoma. And mutations in the optineurin gene are causative for normal tension glaucoma. And these mutations directly affect RGCs, which leads to their neurodegeneration and loss. So in particular, the E50K mutation in the optineurin gene results in a severe degenerative phenotype, which facilitates its analysis in vitro. And being a monogenic variant, it simplifies gene editing applications. So as we all know, the CRISPR-Cas9 gene editing techniques have been emerged as important tools exactly to create these models of disease that we can use to study mechanisms of neurodegeneration, for example. So we can use wild type cells where we can introduce mutations, or we can use mutant cells where we can correct at mutations and create our models of disease. We can differentiate them and allowing for future studies for future phenotypic comparisons. So using these, we use exactly CRISPR-Cas9 in our study. So in a glaucoma patient carrying the E50K mutation, this patient shows an alteration in the codon 50. So instead of the sequence of GAG, a glaucoma with this mutation show a sequence of AAG. So to create our model of disease, we used human pluripotent stem cells, wild types, where we introduced the mutation. So we substitute G by A and create our cells carrying the mutation, our model of disease. Likewise, we also use the cells obtained from a patient carrying this mutation. We corrected that mutation and created our isogenic controls for our studies. So then these human pluripotent stem cells, both carrying the optineurin E50K mutation or the respective isogenic controls, were differentiated as 3D retinal organoids or forebrain organoids for the isolation of RGCs and astrocytes respectively. So we started by confirming that both human pluripotent stem cells carrying the E50K mutation or isogenic controls were able to differentiate into mature and pure populations of astrocytes, expressing characteristic markers like GFAP, EPA-CAM, S100 beta, and SOX9. And interestingly, we observed that a reduced number of astrocytes carrying the E50K mutation were positive for GFAP compared to the isogenic controls. And actually, we confirmed the same reduction by Western blot. So at the beginning, it was a surprise for us, this reduction in GFAP, but we confirmed in the literature that actually, although GFAP has been associated as a classical marker of reactivity, recent works have shown that decreased levels of GFAP have been observed in disease-associated astrocytes, for example, in amyotrophic lateral sclerosis. So through Western blot, we also observed increased expression of SOX9 and S100 beta in E50K astrocytes, again, supporting a disease-associated phenotype in these mutant cells. So since autophagy is optineurin is an autophagy receptor, we then analyzed autophagy markers in these cells. So as you can see here on the left, we observe this accumulation, this aggregation of LC3 in the perinuclear region of E50K astrocytes, while the same was not observed in the isogenic control cells. And when we colocalize this LC3 with the lysosomal marker LAMP1, we observed in a reduction in this colocalization in E50K cells, but an increase in wild type cells. So we conclude that glaucoma astrocytes show a misregulated autophagy pathway and impaired autophagosome and lysosome fusion. Then through RNA sequencing, we wanted to analyze transcriptional alterations in these cells. And we observed that a large number of genes were differentially expressed between isogenic control astrocytes and E50K astrocytes. And in particular, for example, MAPT and CYP1B1 which are genes associated with degeneration in dementia and glaucoma, respectively. So, but also other important genes that we observed through RNA sequencing, like aquaporin or APACAM, which are important markers of astrocytes. So then through pathway analysis, we also observed that a large number of genes that were downregulated in E50K astrocytes were associated with the cell cycle and proliferation, which suggests a more senescent phenotype in these cells, similar to what has been observed in other neurodegenerative diseases. And moreover, we also observed that the inflammatory pathway was one of the most upregulated pathways in E50K cells, again, supporting a disease-associated phenotype in these glaucoma astrocytes. So after this characterization, we wanted to analyze the effect of these glaucoma astrocytes over RGCs. So we performed co-cultures of astrocytes and RGCs, and we performed some morphological analysis. So as you can see here on the right, so if we consider RGCs growing alone without astrocytes, we observe that the E50K mutation promotes some degree of toxicity over RGCs by themselves. So they show this reduction in the number of neurites and neurite complexity. When we add control astrocytes, isogenic control astrocytes, we observe the beneficial effect supporting that astrocytes indeed have a role in the homeostasis of RGCs, showing this increased number of primary neurites and neurite complexity. However, when we co-culture the RGCs with E50K astrocytes, we observe a toxic effect. So the E50K astrocytes promote the reduction in the number of primary neurites and in the extension of the neurites in RGCs, in both RGCs carrying the E50K mutation and isogenic control RGCs. Next, through patch clamp analysis, we wanted to analyze whether these cells, these glaucoma cells, have an effect on the excitability of RGCs. So in dark blue, we have co-cultures where both cell types are isogenic controls, astrocytes and RGCs. In light blue, we have co-cultures where we have control astrocytes, but E50K RGCs. In pink, we have E50K astrocytes, but control RGCs. And in red, we have co-culture where both cells are mutant cells carrying the E50K mutation. So as you can see here, after stimulation, we observed that the action potentials were more frequent when either RGCs or astrocytes were carrying the E50K mutation, and even more frequent if we consider the co-culture where both cell types were mutant cells harboring the E50K mutation. So with these results, we conclude that indeed E50K astrocytes in glaucoma are able to induce hyperexcitability in RGCs, together with all the morphological alterations that we observed before. So after that, we wanted to analyze whether this toxic effect was at least in part promoted by the conditioned media, by toxic soluble factors released by E50K astrocytes. So we performed these transwell co-cultures, and we analyzed that although not exactly in the same degree of toxicity that we observed in direct co-cultures, the conditioned media from E50K astrocytes was able to promote a reduction in the soma size and in the number of primary neurites in both control RGCs and E50K RGCs. So analyzing in more detail the factors that were being released in these conditioned media, we use mesoscale discovery, and we observed that among all the factors that we didn't observe alterations comparing to the basal levels. We observed for IL-6 and IL-8 these reduced levels in the conditioned media of E50K astrocytes compared to the increased levels that were present in isogenic control conditioned media. So we started thinking that probably these astrocytes, these E50K astrocytes, lose the capability to release important factors involved in neurosupport, involved in the homeostasis of RGCs. So trying to explore a bit more this effect, we incubated E50K RGCs with IL-6, And we observed that this incubation was able to promote an increase in the soma size and in the number of primary neurites, together with increased expression of several synaptic proteins in E50K RGCs compared to E50K RGCs without treatment. So this again suggests that glaucoma astrocytes lose the capability to release important factors that have a neurosupportive role on RGC homeostasis. So overall, with this study, we conclude that glaucoma astrocytes carrying this optineurin E50K mutation show misregulated autophagy and dysfunctional autophagy pathway, together with increased inflammatory signaling, but decreased cell cycle and proliferation, which are characteristics of disease-associated astrocytes and senescent astrocytes, and they promote indeed degeneration in RGCs. And importantly, we also observe that these glaucoma astrocytes lose the capability to release important neurosupportive factors involved in RGC homeostasis and proper function. So finally, I would like to thank the members in Meyer Lab, in particular to my mentor, Jason Meyer, as well as other collaborators involved in this study, our funding agencies, and once again to Proteintech for the opportunity to share our research. Thank you, and I'm happy to take any questions.
Thank you very much. Another really nice talk. And we do have a couple of questions from the audience, so I'll go straight to those. First, we have one from Sam Patel, who said, lots of work, very cool. How do you collect and prepare your ACM?
Okay, so we collect the ACM, we filter them to make sure that we don't have cell debris there. And then we incubate it, and then we analyze by mesoscale analysis. That's how we prepared for the analysis. For the co-cultures, we have transwells, so we don't collect the media, we just do transwell co-cultures.
Okay, thank you. We have a question from Shane Liddelow. Nice work, Cátia. I appreciated the neurite outgrowth assays, but I'm intrigued if you did a viability stain to validate the toxicity conclusion. Also intrigued over what time frame the effect is occurring, and congratulations on submitting the paper.
Thank you. Sorry, so it's about the timeline of the co-cultures? I couldn't hear everything.
So what time frame was the effect occurring? This is the toxicity effect.
Okay, so we performed these co-cultures for 4 weeks. So because based on our previous work, we have this indication that the mutation promotes this effect on RGCs after 4 weeks in cultures. So we performed these co-cultures with astrocytes for 4 weeks to see the effect of these astrocytes. So we don't know exactly about toxic effect of factors that have a toxic effect. We are more focused on, and we believe that the toxic effect of these astrocytes is promoted by a loss of supportive role. So we think that they lose the capability to release some important factors in the maintenance and the homeostasis of these RGCs. I'm not sure if I'm answering correctly.
Yeah, Shane said great, thanks. We have time for another couple, and there's quite a few coming in. We have one from Marion Schneider, who said, we'd like to know whether you can quantify S11B in your co-cultures.
S11B.
Have I sent that to the wrong person?
We can do that. We didn't analyze that.
But you haven't done so far?
No.
Okay. Final question we have from Charlie Arber, which says, wonderful work. Your LC3 staining is so interesting. Could the cells be secreting their autolysosomes?
Yeah, we didn't analyze that actually. So that's something that we want to proceed to explore a bit more these autophagy pathways involved in these cells. So that's a really interesting question, and that's an interestingly future direction for sure.
You definitely had a lot of data, didn't you, covering a few different aspects. So I guess there's a lot of future work opportunities there for you. So thank you very much. That was another really nice talk, and thank you everyone for sending your questions in. And thank you again to all of our speakers today. I think everyone's talks have been absolutely fantastic. Thank you to the audience for joining in with all of your questions. If anyone does have any additional questions that we didn't get to cover, let us know and we can put you in touch with people. And do continue the conversation on Twitter with the hashtag, which is hashtag PTGNeuro21. So I'm going to pass over back to Becca for some final closing remarks.
Thank you, Sarah. You were really a great chair there. And thank you so much to all our amazing speakers. I learned so much, and the quality of the science and the standard is just absolutely incredible. And thanks to you all for attending. I'm just going to quickly share my screen again, because we have just a couple of announcements about the Neuro Week that we're running at the moment. So yeah, currently at Proteintech, we have our Neuroscience Hub, so you can scan the QR code and follow to the page. And we're also giving away free samples of all these and neuroscience targets below. You can either do that by requesting it online, or if you want to email me at events@ptglab.com, I can organize that for you. And yeah, thank you so much for watching. And again, like I said, thank you so much to all our incredible speakers today. You can see them up here now, and I've left their emails up there as well, and they're more than happy to be contacted afterwards. So yeah, thank you so much for a really great day, and have an excellent rest of your morning or evening, wherever you are in the world. And thanks for making this such an incredible international ECR event. See you later. Have a great day.